Okay, in this unit we looked at the vascular plants and the fungi. So one of the key things that you need to know is the major evolutionary steps in the evolution of the land plants. So in the first unit we looked at the non-vascular plants. Here the liverworts, mosses, and hornworts, and those first seedless, non-vascular plants arose about 475 million years ago. Then the next major evolutionary adaptation that arose was the origin of vascular tissue, about 425 million years ago. And that gave rise to the seedless plants. So these are the lycophytes and the pterophytes. And then about 300 million years ago, seeds evolved. And we have two main classes of seed-bearing plants. We have the gymnosperms and the angiosperms. And the fourth key adaptation that evolved about 200 million years ago, or 150 million years ago, was here. So about 100 million years ago, we had the evolution of flowers. So the angiosperms are referred to as flowering plants. So like I said, the earliest vascular plants date back about 425 million years ago, and the current living plants, a key difference between the vascular and non-vascular plants is that the dominant generation that you see are the sporophytes. And remember, the sporophytes are diploid, right? 2N. And the key adaptation that evolved in these plants were vascular tissues to transport water and minerals from the soil, and the products of photosynthesis through the phloem. And the development of vascular tissue allows for true roots and leaves. Okay, so like I said, in contrast to the bryophytes, the vascular plants, both the seedless and the seed-bearing plants, the dominant generation is the diploid generation, and the gametophytes tend to be very small, independent organisms. So an important feature to know about any organism is the life cycle. So here we have the life cycle of a seedless plant, a fern. So when the spore germinates, it will grow into a gametophyte. This is a small independent plant that is haploid, right? It's 1N, and it is going to generate gametes by mitosis. So inside, on a single gametophyte, you will have antheridia, which produce sperm, and archegonia, which will produce eggs. When that egg is fertilized, you form a zygote, which will form into a sporophyte, and as that sporophyte grows, it will produce spores, on the underside of the leaves by meiosis, because the sporophyte is diploid. And once you form the spores, the life cycle will continue. So the key thing to remember is that in this life cycle called alternation of generations, you are alternating back and forth between a diploid generation and a haploid generation. Okay, as we said, the important evolutionary adaptation that arose in the vascular plants were xylem, which conducts water and minerals from the soil into the plant, and they are made of cells called tracheids, and at maturity the xylem cells are dead, so the functional unit is just the remaining cell wall from the cell, and we have two different types of vessel elements. We have tracheids, and more recently a more highly derived cells called vessel elements are found in angiosperms. The phloem is the vascular system that transports sugars and organic products from photosynthesis, and they consist of living cells called sieve elements. And as I said earlier, the evolution of the vascular tissue allowed for true roots and leaves in plants. So the roots are organs that are anchoring the plants in the soil, and they are able to absorb water and nutrients from the soil. The vascular tissue also allows for the evolution of true leaves. So leaves are where the mesophyll cells containing the chloroplasts are found, and they are capturing the solar energy for photosynthesis. We have two different types of leaves. We have microphylls, which are leaves with a single vein going up the center, and we have megaphylls. These are leaves with a highly branched vascular system. Another type of leaf are called sporophylls. So on sporophylls, you are going to find spores. And in the ferns, those spores are in sporangia that are found in clusters that are called sori. And other plants, such as the lycophytes or the equisetum, have clusters of sporophylls that are at the tips of the branches, and these cone-like structures are called strobili. So most of the seedless vascular plants are what we call homosporous, so they only produce one type of spore that will develop into a gametophyte that makes both antheridia and archegonia, whereas all seed plants are what we call heterosporous. That means you have a separate male gametophyte and female gametophyte. Okay, these heterosporous species have megaspores, which will develop into the female gametophyte, and microspores, which will develop into the male gametophyte. Okay, so the first group of plants that we looked at were the seedless vascular plants, and this consists of two phyla, the most primitive of which are the Lycophyta, which include club mosses, spike mosses, and quillworts. And the second phylum is the pterophyta, which includes ferns, horsetails, and whisk ferns. So the lycophytes, you can see, have a fairly simple structure and predominantly have microphyls. And as if you remember, in class we looked at this organism Lycopodium, which is very similar in structure to what's diagrammed in the picture above. And you can see at the tips of these reproductive stems, you have these clusters of sporophylls in this cone-like structure, and those are the strobili. The pterophytes have somewhat more complicated structures, such as the fern here. You can see that the leaves are more elaborate and broad to more effectively capture sunlight. Another pterophyte is equisetum, or known as horsetail. And equisetum has two kinds of stems. You have the vegetative stem, which has microphyls that will be performing photosynthesis, and you have the reproductive or fertile stems, which have strobili that will bear spores at the base of the sporophyll. And then the final phylum here is Silotum. So Silotum is a whisk fern, which has what we call dichotomous branching, meaning just branches off in pairs. And at the tips of many of these branches, you see small yellow structures. Those structures are the sporangia, where meiosis is taking place to form the spores. Okay, the next group of plants that we looked at were the seed plants. And in this group of plants, the key evolutionary adaptations were the formation of seeds, was the formation of seeds, and the formation of pollen grains, which allows for the plants to reproduce without the presence of lots of water, because the seedless plants still rely on motile sperm cells to fertilize the egg cells. So in the development of the seeds and the pollen grains, we have very highly reduced gametophytes. So the female gametophytes are typically only 8 cells or so, and the male gametophytes are only 2 or 3 cells. So in addition, the gametophytes of seed plants develop inside the tissues of the sporophyte, so this offers extra protection for the gametophyte. So here's a simple diagram of the similarities and differences between the different plant groups. Again, the key thing with the vascular plants is that the sporophyte in both seedless and seed-bearing plants is the dominant generation. But you're still, like the seedless plants, you're still undergoing an alternation of generations with a diploid organism and a haploid organism. in the non-vascular plants and in the vascular plants. A diploid sporophyte and a haploid gametophyte. So the seeds provide an evolutionary advantage over the seedless plants because the seed is developing within the protection of an ovule, and this allows the plant to provide the embryo with both a source of nutrition and a protective coat to aid in survival of the offspring of the plant. So within the female reproductive structures, we have ovules. So ovules are the megasporangium and the megaspore, which will develop into the embryo sac, which is diagrammed here, and you have protective integuments that are tissue from the parental plant. And in gymnosperms, you have megaspores with one integument. In angiosperms, the megaspores have two integuments. The second adaptation that evolved in seed plants were the development of pollen grains. So now those microspores develop into pollen grains, which are a two-celled structure and carries the male sperm cells. So these pollen grains can be distributed either by air or by pollinators, allowing for reproduction without water being present for motile sperm to swim through. So here's a diagram of a gymnosperm ovule. So you can see here you have one integument on the scale of the ovulate cone, and inside there you have an ovule that contains a megaspore, which will undergo 3 rounds of cell divisions and give rise to the female gametophyte, which will include the egg cell. And the pollen grain will land at the opening at the bottom called the micropyle, and a pollen tube will be generated that will grow towards the egg cell, and the sperm cell that's inside that pollen grain will be deposited into the female gametophyte and fuse with the egg cell nucleus to fertilize to now become the embryonic diploid sporophyte. And the remaining cells in that female gametophyte, which are still haploid, continue to grow and divide and accumulate the lipids and proteins that are going to be required to support the germination of the embryo. So the gymnosperms are the most primitive seed plants. So gymno is, the root is naked, and sperm is seed. So these have what we call naked seeds, meaning that the ovules aren't surrounded by additional tissues. So the gymnosperms consist of 4 phyla. The cycads, the most familiar to you would be the sago palms, such as the large one out in front of the Black History archives. Then we have the ginkgo phyta, which is one living species is all that survive, called Ginkgo biloba. Then we have the nettophyta, which include 3 genera, Netum, ephedra, and welwitschia, all of which are adapted for arid climates. And then the largest group are the coniferophyta, which include the pines and firs and redwoods, as seen here. So the next group of plants are the angiosperms. These are the flowering plants, and they have 2 key adaptations. They have flowers, which contain the reproductive structures and can attract pollinators. And then we have fruits, which develop from the tissues of the flower and are used to aid in dispersal of seeds. So the flower is the structure specialized for sexual reproduction, and the basic flower anatomy has 4 whorls that are concentric layers that give rise to different floral organs. We have the sepals. Okay, we have the sepals, which are found on the outermost part of the flower and are green and leafy. Then we have the petals, which form the more attractive part of the flower to attract pollinators such as insects or animals. And then the inner 2 whorls are the male reproductive structure, which is called the stamen, which consists of the filament, and at the tip you have the anther, which is where the microspores arise by meiosis and the pollen grains develop inside the anther. Then you have the carpel, which consists of 3 different structures. You have the ovary, which contains 1 or more ovules. So each ovule contains a megaspore, which will develop into the female gametophyte. Then above the ovary, you have a structure called the style. And at the top of the style, you have a sticky surface called the stigma, which is where the pollen grains will land, and the pollen tubes will grow down the style to the ovules. So if we look at this life cycle of a flowering plant, the seed will germinate and grow into a mature plant, and then one of those stems will turn into a floral meristem, which will generate the flower. Then within the anther, the microspores will undergo, the microsporocytes will undergo meiosis to produce microspores, which will develop into pollen grains. Those pollen grains will land on the stigma, and the pollen tube will grow down the style and to an ovule. And those ovules arise inside the ovary, and within the ovary you have megasporocyte, which will undergo meiosis, and all but one of those four products of meiosis degenerate, so you have only one megaspore per ovule. That megaspore will undergo rounds of division to make different types of cells, the most important ones being the egg cell and the central cell, which you can see has two nuclei. So when that pollen tube grows down and discharges the two sperm cells, so if you go back and look up here, each pollen tube has two sperm cells. One sperm cell will fertilize the egg cell to form the zygote. The other sperm cell will fuse with the central cell nuclei to form a cell that is triploid, and that triploid cell will develop into a nutrient tissue called the endosperm. And the zygote will develop into the embryo. That ovule will then mature into a seed, and the process continues. So now let's look at the anatomy of the seed plants. So in particular, we focused on the angiosperms. So there are two main classes of angiosperms. We have the eudicots. And the monocots, and they differ somewhat in their internal anatomy. So in each case, we have an outer cell layer of epidermis. Then in the middle, we have the cortex, and then we have a specialized cell layer called the endodermis, and those endodermal cells have a waxy band around them that requires that any water taken up by the plant must go through the cells of the endodermis. Then inside that endodermal layer, we now have the vascular tissue. So in the case of dicots, that vascular tissue is arranged with the xylem in a cross-shaped, and between the arms of the crosses, you have clusters of phloem cells. In the monocot root, you have, those are my dogs barking in the background. In the monocot root, you have the epidermis, the cortex, the endodermis, and the vascular tissue is arranged somewhat differently. So the xylem is found in a circular arrangement on the inside of the vascular stele, and the phloem is arranged on the outside in a ring. So in the stems, we also have differences in the structure. So in the dicot stem, you have these structures of vascular bundles with fiber cells, dark staining fiber cells, because they have thickened cell walls, are found on the outer ring, and they provide support for the structural support for the stem. Then inside that, you have the phloem, and then next to that, you have the xylem. And in the center you have parenchyma cells, which are referred to as pith. And on the outside, I forgot to mention, we've got an epidermis, and the small layer of tissue between the epidermis and the vascular bundles is referred to as the cortex, just like it is in the roots. The structure is considerably different in monocots. So you have the epidermis, and then you have parenchyma tissue, which just referred to as ground tissue in this slide, and the vascular bundles, here you can see, are distributed throughout the stem rather than being in a ring. Okay, the structure of the leaves is also different. So the general structure of a leaf is epidermis on the top and bottom. So we have epidermis on the top and bottom, and then we have a layer of mesophyll cells which contain chloroplasts. The first layer, the cells are arranged tightly packed and oriented vertically. This is called the palisade mesophyll. And then underneath that, you've got cells that are in a much looser arrangement with lots of air spaces between them, called the spongy mesophyll. And in the epidermis, you have specialized structures, pores called stomata. So the stomata are flanked by two guard cells, which can regulate the opening and closing of that pore and allows for the exchange of gases. So this is important for photosynthesis because it requires carbon dioxide from the atmosphere and allows oxygen, one of the products of photosynthesis, to diffuse out of the leaf, because if oxygen gets too high, it ends up poisoning the photosynthesis reaction. And then in the leaf, you also have vascular bundles, which are surrounded by a bundle sheath, and you have bundles of xylem and phloem, and on either side, or associated with those vascular bundles, you have these fiber cells, just like in the stem, provide structural support for the leaf. So now let's talk about the fungi. So the fungi have two main forms. They can have single-celled forms, which we generically refer to as yeasts, and we can have multicellular filaments. And some species, depending on environmental conditions, will switch back and forth between a yeast form and a filamentous form. So this very thin filamentous form enhances their ability to absorb nutrients because it maximizes the surface area to volume ratio. Okay, and we refer to the filaments of the fungi as hyphae, and mats of those hyphal filaments are referred to as mycelia. And fungal cell walls are made of a modified carbohydrate called chitin. This is the same carbohydrate that is found in arthropod exoskeletons, so like in insects. It's what makes the exoskeleton of those organisms as well. So most fungi have individual cells that are separated by cell walls between them called septa. And some fungi are called cenocytic. So in the cenocytic fungi, you don't have a septum dividing the cytoplasm between the nuclei, so you have a cytoplasmic mass with hundreds or thousands of nuclei sharing that cytoplasm along the hypha. So here's an example of a septate hypha, and you can see that the nuclei are separated by dividing cell walls, which are referred to as a septum. In the cenocytic hypha, you can see you don't have any cell walls laid down between the nuclei in the hypha. Okay, so fungi are normally haploid, and the sexual reproduction tends to be a very transient stage in the life cycle. So fungi have different mating types. You can think of the mating types as sexes, essentially. There are plus and minus mating types. And fungi use signaling molecules called pheromones for different opposite mating types to attract one another. Okay. There are some shared characteristics in all the life cycles of the fungi, and they involve plasmogamy, which is the union of cytoplasm from two mycelia of opposite mating type. And in most fungi, when you get a fusion of the cytoplasm of those two mating types, they will mix the different mating types in the same mycelium, and therefore we call this a heterokaryon, or different nuclei. So you have a cytoplasm that contains both plus and minus mating type. In some fungi, the haploid nuclei will pair off in two to a cell, so that you have a plus and a minus nucleus in a single cell, separated by a septum from the other cells in the hypha. So we call these dikaryotic, meaning two nuclei. Okay. So to complete sexual reproduction, you have to fuse those nuclei of opposite mating type, and that fusion of the nuclei is called karyogamy. So this diploid phase is very short-lived, because right after the karyogamy, the nuclei will undergo meiosis to produce spores, which will begin a new haploid generation. In asexual reproduction, many fungi can produce spores by mitosis. Remember, the dominant generation or the dominant organism you see for the fungi is haploid, so you're generating those spores by mitosis. Okay, so the phylogeny of the fungi, we will just focus on the fungal phyla that we looked at in class. So the first would be the zygomycetes, and all of these fungal classes are delineated in the type of structure that they use to form their spores. So the zygomycetes utilize a zygospore. Then we have the ascomycetes, such as this cup fungus, and the ascomycetes reproduce using a structure called an ascus, or it's the root for that is sac. So these are commonly referred to as sac fungi. So you get the ascomycetes from the asci that are used to contain the spores. And then finally, we have the basidiomycetes, such as this mushroom or the mushrooms that we have in our groceries. They reproduce using a structure called a basidium, so that's where it gets its name. So here is the life cycle of a zygomycete, Rhizopus. So the spores will germinate and form a mycelium. In the case of asexual reproduction, those hyphae will put up specialized hyphae, and at the tips you will form a sporangium, which will contain the spores. For sexual reproduction, you have two mating types, minus and plus, and they will be attracted to one another by the pheromones, and they will put out these gametangia, which will fuse and form a zygosporangium. And you can see that zygosporangium has both plus and minus nuclei in it, and because it's got multiple of each, we call it heterokaryotic. Then that zygospore will mature into a very tough, resistant structure, and then inside that you will have the karyogamy. So those nuclei will fuse. You have the diploid nuclei. They will undergo meiosis. Then when conditions are favorable, that zygospore will germinate and form a sporangium, which will distribute new spores. The ascomycetes are found in diverse habitats, and as I said, they reproduce using these sac-like asci, and the fruiting bodies, like that cup fungus that was on the former picture, are called ascocarps. So the ascomycetes play diverse roles in the environment. They can be pathogens, decomposers, or symbionts. And ascomycetes reproduce asexually using a structure called a conidium. So conidia are different than sporangia because they do not enclose the spores. The spores just bud off the tips of the hyphae and then can be distributed by air. Okay, this is an example of a life cycle of a ascomycete. So you can see that in the asexual reproduction, the specialized hyphae called conidiophores just divide by mitosis to make chains of spores, which can be distributed by the wind. So in the sexual reproduction, you can have the formation of a dikaryotic hyphae by a mating type spore. So the mating type minus spore can fuse with a mating type plus. The nuclei from the minus mating type can divide, and you're forming dikaryotic hyphae. So you can see you've got plus and minus mating type nuclei separated by septa in this case. So that fusion of the plus and minus mating type is the plasmogamy. Then in the ascus, the specialized tip structure that will contain the spores, the nuclei will fuse in that process of karyogamy, making a diploid nucleus. That will undergo meiosis to form 4 haploid nuclei. Those nuclei then divide once more so that you have 8 ascospores. And then in the mature ascocarp, you have these bundles of asci, each containing 8 spores. Those can then disperse, and you have, again, plus and minus mating types, and the process can continue. Okay, the basidiomycetes include the mushrooms that you're familiar with, as well as puffballs, and some are important plant pathogens. And the basidiomycetes get their name because they reproduce using this specialized structure called a basidium. So if we look at the life structure, or the life cycle of a basidiomycete, such as a mushroom, you have the haploid mycelia will fuse with one another, again in that process of plasmogamy. And you will form a dikaryotic mycelium. And in the case of the basidiomycetes, these dikaryotic mycelia can live for hundreds of years. So when conditions are favorable, they will form a basidiocarp, which is the mushroom with a stem, a cap, and gills underneath that are lined with these little club-like structures, dikaryotic structures called basidia. Within those basidia, the nuclei will fuse in that process of karyogamy, and those diploid nuclei will undergo meiosis. So within that basidium containing the 4 haploid nuclei, they will mature into spores and will be able to be dispersed by the wind. So one thing I hope you've noticed, in each of these fungal life cycles, you have common themes. They all include plasmogamy, they all include karyogamy, and they all include meiosis. The only difference between them is the structure in which the karyogamy takes place. So some fungi do not have a known sexual stage, so we lump these into the group deuteromycetes. So it's not really a sound taxonomic group, because as soon as you can identify the type of sexual reproductive structure they use, they can be put in one of those other fungal groups. So examples of the deuteromycetes would be Aspergillus, So aspergillus, here you see a mycelium. Here, the cluster of filaments. Here, those are the fungal hyphae, and some of those hyphae differentiate into conidiophores, and on those conidiophores, at the tip, you're getting mitotic divisions, which are producing chains of spores. So you can see that here, you can see the hint of a chain of those spores, and they can be then distributed by wind. The other deuteromycete we looked at was penicillium, and so down here you've got the mycelium, and then the conidiophores, those specialized hyphae, can grow up, and here you can very clearly see those chains of spores that are budding off of the tip of the conidiophores. Okay. One important ecological function of fungi is their mutualistic association with plants, which are called mycorrhizae. We have two different types. We have ectomycorrhizal fungi. Ecto means outside, so these fungi don't penetrate the cells of the root, so they form sheaths of hyphae over the root, and they can grow into the extracellular spaces of the root cortex. The second type of mycorrhizal fungi are the arbuscular. They are different in that they actually form a structure inside the cells of the root, which is diagrammed here. So you can see that the fungal hypha extends this specialized structure called a hostorium, and it branches into what is called an arbuscule, and the function of this structure is to allow transfer of nutrients from the fungal hypha, so the fungal hyphae can penetrate into smaller pores of the soil than the plant root can, so it can more efficiently take up nutrients such as phosphate, and it can transfer that phosphate to the plant, and then the plant can support the growth of the fungus by providing it sugar from photosynthesis. So plants that have this association with fungal mycorrhizae tend to have be more at a competitive advantage over those that don't, because they can acquire nutrition more efficiently. So like I said, the most important aspect of this is that it allows the more efficient uptake of phosphate and certain trace minerals into the plants that the fungi are more efficient at acquiring than the plants are. Okay, that's the end of the review. I hope you find it helpful, and if you have any questions, feel free to contact me by email or come and visit me during my office hours from 2 to 4 on Monday and Tuesday and Wednesday.